Aging. Explained by Iris, Frisia, and Izzy. Question 1.
What causes aging? Although there are a number of factors that contribute to what we call aging, we are going to focus on the genetic aspects.
There are two types of genes that cause aging and the shortening of lifespans. Genes that cause specific diseases associated with aging, and genes that code for the body system failures that produce the symptoms of aging. Several diseases are commonly associated with aging and old age, including Alzheimer's, cardiovascular diseases, arthritis, cataracts, osteoporosis, and type 2 diabetes. All of these diseases accelerated the aging process by drastically shortening lifespans. Genetics plays a large role in determining an individual's vulnerability to age-associated diseases.
Alzheimer's disease, the degradation of the brain that causes progressive mental deterioration in the middle or old age, does not yet have a definitive cause.
However, there are several genes that have been found to be associated with the disease: the APP, PSEN1, PSEN2, and the APOE genes.
Mutations in the first three cause early-onset Alzheimer's by allowing for a toxic buildup of amyloid beta peptide, which clumps in the brain and leads to the death of nerve cells and the resultant symptoms.
Late-onset Alzheimer's is associated with the apolipoprotein E gene, specifically a variant called the E4 allele, which increases the likelihood of developing the disease. Cardiovascular disease can also be attributed to genetic factors. Mutations in many genes that code for the protein components of the sarcomere, a structural unit in muscles, have been shown to contribute to cardiovascular disease and heart failure.
Although many other factors lead to the development of heart disease, certain genes put individuals at much higher risk of getting the disease or the factors that make it more prevalent.
Diabetes is one of the factors that can lead to heart disease, and it is also linked to specific genes, the TCF7L2, ABCC8, GLUT2T, and the GCGR genes. Variants in these genes seem to be linked to the onset of type 2 diabetes.
These are just three of the disorders generally associated with the aging process. However, arthritis, osteoporosis, cataracts, and more are also thought to be partially caused by mutations in specific genes.
Some diseases actually cause the symptoms of aging instead of just shortening lifespan. There are several diseases that cause premature aging, such as Werner syndrome, Cockayne syndrome, and Hutchinson-Gilford syndrome. Werner syndrome is caused by defects in the WRN gene, which disrupts the normal production of DNA helicase and therefore the transcription and DNA repair processes. Cockayne syndrome is caused by a gene involved with transcription and DNA repair as well, a gene called ERCC8. Hutchinson-Gilford syndrome differs slightly in that it is associated with the LMNA gene, which codes for components of the lamina, a layer of the inner nuclear membrane. The genes that cause these premature aging diseases most likely contribute to the causes of regular aging because they cause symptoms like hair graying and loss, wrinkling, skin atrophy, loss of bone and muscle mass, and age-associated diseases.
Other genes have been investigated as possible causes of aging as well. The DAF2 gene is a gene that controls a pathway for other genes, which then regulate physiological processes throughout life, processes that degrade as an individual ages. Typically, DAF2 deactivates DAF16, which codes for a transcription factor that activates stress resistance genes and is generally associated with longevity. Similar longevity genes are GATA transcription factors, which control other genes that are expressed differently in the young and the old. These factors become more unbalanced as the body ages, disrupt genetic pathways. When the system starts to degrade, its previously useful functions leads to an accumulation of damage to the circuits that work to express certain genes and therefore the aging process.
The SIRT1 gene is another that has been associated with aging. This gene has been found to influence DNA repair and to suppress certain genes. It has the ability to regulate the cell by controlling waste production. Smaller amounts of waste allow the cell to work better for longer. The failure of these genes to maintain cellular regulation, which is needed for reactions to stressors and therefore longevity, results in the quicker breakdown of cells and therefore aging symptoms.
So all of this information leads us to ask the question, what research is being done? In addition to studying the genomes of human individuals with various age-associated diseases, scientists have looked into the aging genes of other species such as yeast, worms, and mice. This is because many believe that looking into the genetics behind diseases is useless. Finding a genetic solution for one disease will just open the path for another age-associated disease or create other health-related consequences.
Scientists have been studying yeast cells in order to determine genes associated with aging. They were able to isolate two genes, GRE3 and ADH2, that help to control the gene expression in aged specimens of yeast. They discovered that turning off these two genes causes the gene expression in the old yeast to revert to the expression of a young cell. Using the metabolic transformation algorithm that allowed these genes to be determined, sets of genes that cause the expression differences in old and young humans can be isolated and even turned on to prevent aging. Worms have also been a source of interest in terms of the genetics of aging. Studies of the genetics of the C. elegans roundworm allowed scientists to isolate the GATA factors known to contribute to aging. They discovered that the ELT3, 5, and 6 genes were specific GATA transcription factors that controlled the expression of genes in young and old worms, therefore causing aging. When these genes were restricted in the worm specimens, the worms lived up to 50% longer, indicating that these genes were the ones causing it. This discovery set the stage for further investigation into the effect of GATA factors on human aging.
Mice have played a key role in this research because they respond to genetic manipulation and reproduce easily. Scientists have isolated genes related to aging in mice through various types of this genetic manipulation. They have also used mice as a way to look into possible cures for the aging process. And that brings us to our third question. Can and should the aging process be stopped or reversed?
New drugs have been researched and developed in order to attempt to slow the aging process, and various drugs have been shown to increase lifespan by affecting certain genes. So far, nothing has been discovered that can completely stop the process. TOR, IGF, and AMPK are three genes that affect human lifespan. The decrease of TOR and IGF expression and the activation of AMPK are two genetic changes that could ultimately extend the human lifespan.
Metformin is one of the drugs that has been investigated for its effects on the aging process. It is typically used as medication for type 2 diabetes, but it has been found to have certain anti-aging properties. So far, it has been shown to prolong life in both mice and roundworms. It is thought to affect aging by activating the dormant AMPK gene, which slows the aging of body systems and induces autophagy, a process in which old and damaged parts of cells are degraded. The buildup of these old cellular components contributes to age-associated diseases and system failures, but the activation of the AMPK gene is able to combat this process and therefore delay the onset of aging.
Recall the progress of anti-aging experiments on mice. Autophagy can also be induced by the restriction of certain genetic signaling pathways like the TOR and insulin growth factor pathways. In mice, a drug called rapamycin was used to inhibit the effects of the mTOR pathway. This pathway is necessary for healthy growth in young specimens, but has a more negative effect in older individuals by causing aging symptoms. In mice, the suppression of the mTOR pathway extended lifespan and delayed the onset of age-related illnesses. A decrease in this pathway, or in the IGF pathway, which also contributes to cellular autophagy, can allow for longer life and less age-associated disease, which would slow the aging process.
Another method of aging prevention that does not involve pharmaceuticals is the use of gene therapy, specifically telomerase therapy. Telomeres are regions of repetitive nucleotide sequences at the ends of chromatids which protect the chromosome from deterioration. A little bit of telomere length is lost each time the cell divides, which limits the number of times a cell can divide before it destroys itself. Whoa. Researchers are attempting to lengthen the telomeres in order to possibly extend lifespans. By using non-replicating or non-pathogenic viruses as a vector to insert additional telomerase genes into human cells, telomere length can be extended by as much as 1,000 nucleotides. This could potentially add many years of life to human lifespans. Further investigation must be done to see how effective telomerase therapy could be in effective aging genetics.
In conclusion, the manipulation of genetics could be the next big thing, but there are some risks. Control over aging genes can have drastic effects on one's fertility early in life. However, aging genes are extremely promising, and the idea of prolonging life even further is a topic that has just been studied in the last 20 years. These risks, among other factors, cause hesitation on whether or not creating medication that stops aging is a good idea. Many believe that because it is defying nature, getting rid of the aging process would be considered inhumane and preposterous. Halting aging isn't about playing God. It's about reducing the amount of children without parents, lowering the death toll of devastating diseases, and allowing for more individuals to walk out of hospitals cured. With hard work and much more research, nobody would ever have to watch as their parents lose their memory, watch as the person they used to know fade into an empty shell that slightly resembles their family member. If you had the opportunity to save thousands of lives at your fingertips, would you?